My name is Charles Warwick, a postdoctoral researcher at the University of Pittsburgh Center for Pain Research. With the help of our co-authors, we have published the article MrgprdCre lineage neurons mediate optogenetic allodynia through an emergent polysynaptic circuit. In this work, we investigate the role of the genetically defined primary afferent population MrgPRD in the development of neuropathic pain and the spinal mechanisms which transform an innocuous stimulus into one which drives behavioral avoidance. Traditionally, cutaneous C fibers were presumed to be nociceptors, as they respond robustly to noxious stimuli. However, the IV4-binding non-peptidergic C fiber afferent population, which is marked by the adult expression of the gene MrgPRD, also responds to innocuous levels of mechanical input, thus raising the question of whether activity in this population represents true nociception. To study this population in a more nuanced fashion, we utilize the MrgprdCre, which labels these non-peptidergic C fibers, and when crossed with a channel rhodopsin, allows us to specifically interrogate their role in nociception. Surprisingly, we found that in contrast to the TRPV1-Cre, the MrgprdCre lineage cells did not produce robust, traditionally pain-associated behavioral withdrawals to the optogenetic stimulation of the hindpaw with blue light. Furthermore, this population did not drive behavioral aversion in a real-time place aversion assay, suggesting that non-peptidergic activity per se in a naive animal is not sufficient to drive behavioral aversion. However, when these animals are subjected to a spared nerve injury, optogenic activation of this population now drives behavioral aversion, likely representing a form of allodynia being driven by the previously innocuous optogenic activity in the MrgPRD population. We next sought to determine the mechanism of action for this striking phenomena, and first examined peripheral mechanisms of change. Interestingly, there were no changes after SNI in the labeled primary afferent population in regards to either the expression of the channel rhodopsin or the optogenic response in intracellularly recorded MrgprdCre afferents. We therefore assumed there must be a central mechanism to explain the development of this optogenic allodynia. We then performed patch clamp recordings of lamina 1 spinal parabrachial neurons using the semi-intact ex vivo preparation. We found that after SNI, a greater proportion of spinal parabrachial projection neurons were activated by either innocuous brush or optogenic activation of the MrgprdCre afferents, suggesting that changes after SNI permit more of the MrgprdCre input to reach lamina 1. Consistent with this hypothesis, we also discovered that after SNI, the latency from optogenic stimulation to the onset of the EPSC at the projection neuron was significantly increased, suggesting that the MrgprdCre afferent input is being relayed to lamina 1 by an emergent polysynaptic circuit that is normally silent in naive conditions. Together, these results suggest that under naive conditions, MrgPRD input is behaviorally innocuous due to the spinal inhibition, which limits its contribution to spinal parabrachial projection neuron activity, which is required for non-reflexive avoidance behaviors. However, after injury, when the MrgPRD input becomes behaviorally aversive, we suggest that this polysynaptic circuit becomes active and permits more of the MrgPRD input to reach the spinal output neurons, thus contributing to the enhanced output of spinal parabrachial projection neurons after injury. Thank you.